Now we'll move on to the next speaker, Liran Shlush from the Weizmann Institute, telling us about early diagnosis of myeloid malignancies. Thank you, Liran.
Thank you, Ronit, and thanks a lot for the organizers for the opportunity to speak today. Let's start. So acute myeloid leukemia is one of the most devastating leukemias and one of the devastating cancer in general. It is a disease of stem cells. In this case, stem cells acquire mutations, and instead of doing their function of differentiation, stem cells, hematopoietic stem cells, are supposed to create the entire blood system. Stem cells have differentiation block for reasons we do not fully understand all of the time. And because of this block, they get signal to continuously divide and proliferate, but they cannot differentiate. So the stem cell pool is expanding to the extent where at some point stem cells inhibit normal stem cells, and we get cytopenias, which are the presenting symptoms of acute leukemia. Acute leukemia, as I said, is a devastating disease with more than 70% mortality rate over the last 4 decades. The mortality rate haven't changed much, and the reasons for that are because the population is continuously aging, and although we do produce some innovative treatments with the aging population increased rate, we still get the same rate of mortality in the last 40 years. We stumbled into a phenomena in stem cells and the identification of pre-leukemic stem cells by the following experiment. So we sequenced AML cells and T cells from the same patients, and we found that both the AML cells and the T cells share an identical mutation in a gene called DNMT3A. The name of the gene is not important because this is true for many other gene mutations. The fact that that AML cells and T cells share the exact same mutation suggests that there is an earlier cell, which we termed pre-leukemic stem cell, that carry an initiating mutation. This initiating mutation does not cause any effect on differentiation because the stem cells can still differentiate and produce T cells, B cells, and any mature blood cells. However, at some point, these stem cells acquire the next hit, and that will cause the differentiation block. So this early step of pre-leukemic stem cells, which we identified by others also around the same time, is probably the early stages of acute myeloid leukemia. And as we showed later, and I will show you today, these early stages start many, many years before leukemia is actually presenting itself, which gives us an hope that leukemia might be even preventable if we identify these pre-leukemic stem cells and target them before leukemia evolves. An interesting fact about stem cells and aging. So this is a study that looked on single cell stem cells from a healthy individual. And you can see that when we are born, there are almost 0 stem cells, and then they expand during the gestational period. And then we reach around 100,000 stem cells by our maturity. And then this number is slightly increasing with time. So hematopoietic stem cells number are increasing as we age for reasons we do not fully understand. These stem cells do not increase in an equal way. So what we described and others described also is that hematopoietic stem cells, one of them at some point in the aging process acquire somatic mutations. Here you can see in X the red cells, and these somatic mutations lead to clonal expansions of specific hematopoietic stem cells. This clonal expansion is termed age-related clonal hematopoiesis, or ARCH, and this is the preceding step of acute myeloid leukemia, but also of many other myeloid malignancies. And people have termed this clonal hematopoiesis ARCH if 2% of the peripheral blood cells of healthy individual carry somatic mutations, which we can also see in leukemia patients. So 2% of cells that carry a mutation define clonal hematopoiesis. And this phenomena is associated with leukemia because it's the same mutations that cause this clonal hematopoiesis and cause early leukemia. People have demonstrated, these 3 pivotal studies have demonstrated that the frequency of clonal hematopoiesis, or ARCH, in the general population can reach 10 to 15% in the elderly population. That means that in this audience there are people carrying somatic mutations in their peripheral blood, and in most of the cases they will never develop leukemia. These cells will dwell in their bodies. If they will live long enough, then the frequency is going up and up. And at some point, some of them will develop leukemia, while others will die due to other reasons. These are the mutations that are associated with this phenomena. All of these mutations are also described in acute myeloid leukemia and other myeloid malignancies. So it's mainly epigenetic modifiers like methylation modifiers and other splicing factors. So the things that are related with the changes of many, many different genes. If you change the methylation, in general, you will change the gene expression of many, many different genes, and the mechanism of why these mutations cause this clonal expansion over time remain unknown and are a big subject of study in our lab. Sorry, oops, it went to the end. You'll have to. Okay. An interesting fact about this clonal hematopoiesis is that it's not just related to acute leukemia and the evolution of leukemia, it is also associated with hyperinflammatory states. In this case, you can see how deficiency in TET2, is present in hematopoietic stem cell, will be present in all mature cells. In this case, what happens is that monocytes that carry such TET2 deficiency or TET2 mutations go to atherosclerotic plaques and secrete IL-1 beta. This IL-1 beta causes the absorption of cholesterol into the plaque and causes increased plaque size. This is why this phenomena of clonal hematopoiesis was not just associated with leukemia, is also associated with all-cause mortality and specifically heart diseases due to the effect of mature cells carrying these mutations on different tissues in the body. And recently there are nice papers showing that T cells carrying this TET2 deficiency are also hyperactive when you're using them as CAR T cells. So using actually TET2 deficient CAR T cells is actually beneficial for patients. There was a nice Nature paper about that. So this phenomena of age-related clonal hematopoiesis can cause cardiovascular disease and other diseases. However, in our lab, we are interested in this process of evolution from clonal hematopoiesis to a true pre-leukemic state that will lead to different myeloid and lymphoid malignancies. So you have to understand that most of the myeloid malignancies start from this early clonal hematopoiesis that comes many, many years before we get the actual disease of leukemia. So this is the focus of our lab. And the big question of our lab is, can we diagnose and treat leukemia earlier? So instead of waiting for the leukemia to present itself, can we identify high-risk individuals carrying mutations many years before they develop leukemia and then offer them preventive therapies, not to wait for the actual tumor to evolve? So this is the study we took 4 or 5 years ago. We started it. We went to a cohort called the EPIC cohort, which is the European Prospective Investigation into Cancer and Nutrition. This cohort includes 500,000 people that were collected all over Europe. The blood was collected in the early 90s, and then they were followed for the last 30 years. Every time they developed any cancer, it was recorded. So now any scientist can go to this cohort and ask for the blood that was collected many years before cancer was diagnosed. We did that for all the AML cases, 124 in this case, and then also 677 controls that were matched by age and by the region where they were collected. So now we have in hand the blood of individuals who develop leukemia, but we have their blood 8 years on average before they develop leukemia. And then we have the same blood from controls who never develop any myeloid malignancy. One can do that for any other cancer. It's just that the only tissue available is the blood, so one can look on plasma, serum, and blood cells. So, and this study was in huge collaboration together with George Vassiliou from Cambridge University, who helped us in re-evaluating all our results and validating our results on a different cohort that he assembled. What we did was we used error-corrected targeted sequencing, which we improved and reduced costs to be able to sequence very deep at average depths of 5,000x of 261 AML-related genes and identify early mutations already many years before leukemia is diagnosed. This is briefly what next-generation sequencing is and what is variant allele frequency, which is a term that you will hear a lot during this talk. So next-generation sequencing is piling up reads of sequence, much like Sanger sequencing, just you have a lot of them covering the same genomic region. And every time you have a mutation, it is called, like here, A, and then you can calculate the number of reads that carry a mutation versus the total number of reads, and this gives you the burden of the mutation, or even the burden of the pre-tumor or tumor. So a VAF of 2 out of 18 is 11%. Most cancer mutations are heterozygous. They do not occur at the exact same positions in the two alleles. So you can calculate from that what is the percentage of cells carrying this mutation, and it will be double. So 22% of cells in this case are carrying this somatic event. And this is an example for a homozygous mutation, which is most probably germline and not somatic. So this is the difference between mutations acquired as we are aging versus mutations we were born with, germline mutations. And these are the first findings from our study. So 80% of the individuals who actually developed AML already carry mutations that are known to be related to leukemia 8 years before diagnosis. You can see that as we age more, or as people age more, carriers will carry these mutations. At young age, actually, very few AML cases actually carry these somatic mutations. And the reason for that is that young age leukemia is driven mainly by translocations of transcription factors, and we cannot detect these translocations by the methods we have used. This is why we couldn't identify early leukemia cases among the young population. So our methods for early detection are mainly suitable for the elderly population. In these cases, they develop leukemia mainly from point mutations in the genes that I've listed before. And you can see that the general population also has a very high carrier rate of around 20 to 30%, and this is increasing with age. And this is the challenge here, because if so many individuals carry mutations and will never develop leukemia, how we can identify the ones that are truly going to develop acute myeloid leukemia? So this is exactly what we tried to do. And we look now on the variant allele frequency, which I just described before, and we found that the individuals that are destined to develop leukemia have much bigger clones than the general population. That means that they carry more cells in their blood are carrying mutations, and probably the burden of this clone size is reflecting the longer time the disease evolved in their body, because as you will see soon, these early stages of leukemia grow in a steady state, in a steady pace, and the longer you have these pre-leukemic stem cells in your body, the bigger the clone will be, and probably some epigenetic evolution is happening even without the accumulation of the next set of mutations, which lead to the more aggressiveness of these early stages of the tumor. And this is what I described about the growth rate. So on the left side, you can see the controls. In some of the cases, we had the same individual measured the variant allele frequency at least 2 time points, and controls look like they are maybe shallower or not as steep as growth rate as the pre-AML cases. However, when you correct for the initial clone size, because the initial clone size was much bigger in the pre-AML cases, you can see that the growth rate of the controls and the pre-AMLs is actually identical. And maybe specific mutations, because each color here, it defines the specific mutations, have different pace. However, in general, if you're a control, if you're pre-AML, the growth rate of your pre-cancer is identical, not as much as some people might think that if you're going to develop cancer, your cells are dividing more rapidly. Not according to our data, at least in the early stages of the tumor, the cells are growing at the same rate. It depends how long you're carrying these mutations probably in your body and what type of mutations you carry, because some mutations have intrinsic properties that give them stronger oncogenesis properties, and probably under specific microenvironments they will grow better. So this can go now to much finer resolution to look at the specific mutation. However, we couldn't do that in this small cohort. Then the next step was to try to see whether specific genes are differentially represented between the cases who develop leukemia and the controls. And what we found that several genes like SRSF2 and U2AF1, TP53, RUNX1, IDH2, and IDH1 were almost exclusively found in the individuals who eventually develop leukemia. So there are some genes that if you find them in healthy individuals, that means that almost 100% they will develop leukemia in their lifetime. That means in around 8 years. Which means that if they are 85 and they have stage 4 congestive heart failure, they will probably die from that reason and not from leukemia, so we will not intervene. However, if an individual is 50 years old and we can find one of these mutations, he will probably develop AML. And all of these cases, you have to understand, I don't want to put anyone here in panic, they all died. Leukemia is a deadly disease. All of our AML cases, when we looked 20, 30 years after the cohort was collected, they were all dead. And we know that SRSF2, P53, U2F1 AMLs are very deadly. There is more than 90% mortality rate in a year. So clearly, an early intervention that might prevent the actual diagnosis of the disease can be beneficial, or that's at least our opinion. Okay, so to summarize these parts, we can see that pre-AML cases have bigger clones. I didn't show you, but they usually have more than one clone because we found more than one mutations in their blood. They have larger clones. They will have specific mutations that have high penetrance, like the IDH1, IDH2, which is interesting mutations because there are specific drugs that can target these mutations and maybe can be given very safely to healthy individuals who carry these mutations. These are orally prescribed drugs. Other mutations that have high penetrance are the splicing mutations and p53. We think that the leukemic stage of the disease is probably very short and happens in months or weeks, and this is the case where you get the block in differentiation and expansion of progenitors, and the controls just look different. We think that there is a therapeutic window in this stage, and we want to try and treat patients in this stage in the future. Okay, so we were able to produce a mathematical model that can predict leukemia. So if we take the mutation pattern of a healthy individual, we can predict whether or not he will develop leukemia at relatively good accuracy. However, the sensitivity is not perfect, and also the specificity is not good enough. So we wanted to find ways to try to enrich the population that actually is going to develop leukemia if we're going to do prospective studies and try to identify healthy individuals carrying mutations. To this end, we looked at the blood parameters of the patients in our cohort, and the only parameter that was different between the pre-AML cases and the controls many, many years before AML diagnosis was the red blood cell distribution widths. This parameter, if you can see here from a patient of mine, is something that everyone can find in his blood test. Don't do it now and see that you're high. It's just going to distress you. But it's part of the normal CBC blood test that everyone takes. So we can use that parameter to screen large population because it was increased in individuals who developed AML. So what is RDW? RDW is the distribution of the size of the red blood cells. So it's not whether they are big or small, it's just how wide the distribution of their sizes is in the population of all red blood cells. And as you can see, this parameter, RDW, is increasing with age, specifically in males, and look a little bit different in females because of iron deficiency anemia. There are many, many reasons for increased RDW across the population, and we are talking about increased RDW without any other change in the blood parameters. So completely normal hemoglobin and all other parameters are normal, just increased RDW without anything else. And this is how it looks on the Clalit data. So we have access with the Amos Tanai group and the Clalit group to all CBC data of 4.5 million people, and you can see that increased RDW is very common. However, when you look on increased RDW that is not due to iron deficiency anemia, which is here, and is located outside of the borders of anemia, which is the big circle there, you can see that there are many pre-AML cases with increased RDW. This is from the Israeli data, so we have validated the results of the EPIC in the bigger cohort of the Israeli Clalit data. Then we went prospectively and looked for individuals who have increased RDW above 15 in a cohort from Ichilov. We looked for 300 people with increased RDW out of 36,000 healthy individuals that all they had was increased RDW. And then we looked on normal people with normal RDW. And we found that in the increased RDW, there was much bigger variant allele frequency. And this is now prospectively in healthy cohort of people who never developed AML. And besides having bigger variant allele frequency in the group of the high RDW, they also had way more high-risk mutations, specifically in SRSF2, IDH1, and IDH2, and U2F1, that were found only in the group of people with increased RDW. So now we have a measure to enrich for the high-risk patients, and we think that in this group we might be able to intervene as we are developing drugs that can target splicing mutations, and there are already drugs available to target IDH1 and IDH2 mutations. Okay, so the last part of this talk, a nice story of a student of mine, Tzach. So he was looking on mutations in SRSF2 and SXL1, which are again high-risk mutations, and he found that the position of where the mutation appears is non-random. He found that specific mutations are way more frequent, as you can see here. So the x-axis is the position across the gene, and the y-axis is the frequency of specific mutations. And some mutations are very frequently found in specific individuals. And this begged for why this is happening, why specific mutations occur at a very specific position. So what Tzach found was that these mutations are in these genes, KELAR, SXL1, and SRSF2, and they are very, very frequent. And when he looked on the actual sequences of these mutations, they were all deletions. And very interestingly, these deletions always occur identical sequences at the border of the deletion, and what probably happens, or not probably because Tzach proved it, is that the DNA breaks in the middle, and then there is a machinery that combines the two homologies and gets rid of one of the homology, and everything is between. And this is why you get always the exact same size of deletion in all of these cases, and this is true for the KLR deletion, the SXL1, and the SRSF2 deletions. They are all occurring by the same mechanism. And the mechanism is probably by double strand break, and then the ligation of the microhomologies by a system that we do not fully understand its enzymatic machinery yet. We are studying that right now in the lab. So this is what Tzach did to prove that. He created double strand breaks outside of the microhomologies, inside the microhomology, and in between. And you can see the orange bar. He was able to recapitulate the human mutation in vitro by introducing double strand break exactly between the microhomologies, and then you get the annealing of the two microhomologies and creating these recurrent deletions, which is a very interesting finding because it tells us the story about the very early mutations in leukemias, the pre-leukemic mutations, and how they are created. They are created by double strand breaks occurring at a very specific positions and a peculiar DNA repair machinery that will always create the same deletion, which will eventually lead to leukemia, which almost makes no sense. Why the human body will behave like that? Why will create always the same repair that will create the exact same mutations? So we are studying this now in the lab. I just want to show you that these mutations are very common in other diseases, in myelofibrosis, not just in AML, and they are caused due to this repair machinery. We then tried to do the same experiment in another gene called DNMT3A, where you never see this type of deletions. And although we can create these green MMEJ deletions, these microhomology deletions, these deletions are never found in humans. So this is specific to specific genes, probably specific to specific positions of the human genome. And then this is the machineries that are supposedly correcting the DNA. And we're looking here on two options of what is happening. Either it's due to SSA and microhomology repair, or due to alternative end joining. We still do not know which of these pathways is responsible for these deletions in the myeloid malignancies. But what we're doing now is we're inhibiting different enzymes from this pathway, and we can see that we can, by inhibiting the non-homologous end joining, we can delete eliminate specific insertions, and by inhibiting the MMEJ, we can delete other, but we can never delete the canonical mutations that we found, and we now think that it's mediated by the SSA pathway. Okay, so to summarize my talk, we described the early stages of leukemia. We are studying in the lab the aging of the bone marrow, how it affects the growth rate of these different mutations, and maybe the effect on the bone marrow can explain why specific individuals develop leukemia and others don't. We are studying the mechanisms of high-risk mutations, why specific mutations occur to begin with. We don't think that mutations occur randomly. There is a reason for everything. And we're doing a lot of drug development to try and target these high-risk mutations and prevent leukemia at the end. I would like to thank the people who did all the work. So Sagi did the work on the EPIC cohort together with Maurice, Grace, and George, and other people from the lab. And Tzach, Feldman, and Yoni did most of the work on the deletion mechanism. And thank you for listening today.